So as you probably learned a lot of detail for this particular topic already. So this is the last year I've given a similar topic, and I kind of present to you why FAST or Allison convinced me took this project 2 years ago. So there's many reasons for, but one particular reason is highlighted here, and you probably have seen many different versions of this particular diagram. And so I used a different color, which I inherited from Dr. Baudel, which our student of his lab back to many years ago in Texas. So the red color means the gene only expressed from the mother's chromosome, and the blue color is the gene only expressed from the father's chromosome. Then you heard a lot of antisense. So you heard a lot about the animal model for since yesterday and today too. So, but the one particular point that I try to make here is, yeah, overall study animal model is very valuable. However, there is a clear difference. Allison told you already, Quincy is not mice and mice is not human. So therefore, if you truly say, okay, I want to study it in the human brain, that is have to find an alternative way to do it. And luckily, we have the new technology just developed about 10 years ago, and a new brain organoid just 5 years ago became popular. So the project, Allison convinced me to take it to say, hey, Yong-Hu, why you can, because you cross both research and the patient contact, maybe you can help to establish this kind of IPSC cell line, which is basically the induced pluripotency stem cell. So we set up a goal aim one is create a larger panel of Angelman iPSC cell from patient. Many of you in the audience I have interacted with, and over the last year, many years ago, and even back to Duke, and maybe I have seen you in the clinic. So we cover all the genotype. Second is really is a fast interest is to figure out how to distribute the cell line to any investigator in the country, in the world, including farmers, so people can really working hard and use the cell line to study them. So as you are familiar with this kind of slide, so we try to, the plan is try to create 20 cell line for each genotype. Now here we list about 5 genotypes. First one, you're all familiar, it's a larger deletion with 70%. The second is a UP3 point mutation. And the third is UPD. And the fourth is imprinting defect. And the last group we add in, we call it UP3 gain of function mutation. When Allison talked to me, said, okay, I don't want any family in the audience or in our community be left out for this particular research project. So I think she had a very, very good point. That's the whole purpose is every genotype is covered. Now, of course, we include a normal control. So this is a very simple diagram. How do we do this work? Some of you in the audience probably know I talked to you over the Zoom meeting and explain you in the detail, but a very simple slide here. So we took the blood from your kids with Angelman confirmed diagnosis, and we using the cell in the blood, and you transfer the cell, use the Federal Express here, come to the Yale for 24 hours. I know it has to be quite difficult. We started this project during the middle of the peak of a COVID time. And thank you so much, every one of you contact us and also making very, very special arrangement during the, I know it's so difficult over the time, and you have to bring the kids to the clinic and get a sample collected and then the shipping to us. But so far, everything worked out really, really well. So I want to, again, here to thank everyone you contact us, participant for this project. So basically, we take the cell and we go to the Yale Stem Cell Laboratory, which is one of the first or largest stem cell in the country, probably in the world. Then they have a lot of experience to using very, very special technique. The techniques actually got a Nobel Prize a few years ago, to converting the blood cell to the stem cell, which, let me see if probably can use the point here. So now you can see after many, many days, and so in the petri dish to work, so you can see the blood cell became something like this, which we oftentimes say, oh, okay, the stem cell is come along. So once you get a stem cell, now you really say, oh, can we let the stem cell differentiate to other type of cell? Of course, for Angelman, we're really interesting to differentiate this stem cell to the neuron, the brain cell. Now you really say, oh, now since I have the neuron from individuals, Angelman patient, so we really can study brain cell from Angelman for understanding what we call the mechanism or test and treatment. So that's the whole purpose is. So here's the update, I would say, progress so far. Since I think we started project roughly last October, we have some 4 or 5 months delay due to the initial sort of peak of the COVID time. So we are getting started about last October, November. So far, we have generated 12 cell lines. So you can see this is the summary for each genotype. We got almost every genotype covered, true. You can see we have a deletion, we have 3 lines, 2 male, 1 female. We have the UB3 mutation, also 3 cases, 2 female, 1 male. Then we have a UPD imprinting mutation. We even got 1 family, it's a gain of function mutation. We only so far only got 1 of the normal control. We know why we're behind. I think many of you, we're asking whether sibling can join this project. You are very, very generous, say, yeah, we can. However, we are kind of somewhat limited for the RRB by Yale because when you have an older sibling, particular teenager sibling waiting to give them the blood, and we need to separate the form, called ascend form. And due to this kind of COVID Zoom meeting time, and we slow this process, we just get all this form approval very, very soon, next couple of weeks. So then we come back again asking your participant as a control for some of family already committed to. So far, we already generated some of the cell line. After we generate the cell line, we actually have to do more work. The general production cell line is just a simple step. Because when you generate cell lines, because there's a lot of manipulation going on, you really want to make sure this cell line in the good quality and the good standing for other kind of downstream application. So we have to check out, okay, because we use a very special virus, called the Sendai virus, for induce this ESC cell. So we have to make sure the ESC cell is virus free of the virus. So we do that work first. After one, because the limitation some of this technique, when you program this sort of somatic cell to the stem cell, sometimes can cause some unexpected event at the molecular level. So we want to make sure that happen. Will not happen, but didn't happen for the cell line we generated. If that's the case, we may say, okay, this cell line, this particular clone may not be useful. So we are very, very careful, kind of do all this additional very comprehensive genetic sort of workup. Make sure the cell we generate from your kids, then to really recapitulate first the original mutation will keep it. It's a mutation, it's a point mutation or deletion or UPD and imprinting defect or maintain in the cell line. Second, we're not induce additional sort of other unexpected genetic mistake in the cell line. So then the other people from the other investigator may cause the confusion using this cell line. Of course, we will do best we can, and still sometimes still can cause some problem for particular line or clone. So that's why we want to generate enough number of cell line. I think 3 is a minimum. For a few number of the class, I end up saying, okay, if that's 4 or 5, we open to generate more than 3. It's the best for the community if we have more than 3 a particular type. So the good news is that even with still continued progress, we already distributed some of cell line to some investigator. That's a farmer contact us. We will very quickly also deliver this cell line to the farmer once all the paper will get done. The academic investigator, the paper was much simple, and for the farmer, we just need the extra paperwork, but eventually we'll free delivered to them for the use of the cell line. So that's the project one, but again, we're taking this opportunity to thank you all the family participants, and we will continue engaging with you because we want to collect another 10 cell line, and I know some of you waiting and for us kind of getting to the next step. We will be in touch very soon. So the second project is switch the gear to the mice, and as Allison earlier also alluded to, we taking the project, Allison doing very, very good job kind of to convince me to take these two quite challenging projects. The second project is, okay, let's look at this genetic, so the mouse model we have right now so far. So as you heard many talk or read many publications, there are many mouse model available. So this is more detailed. So this top panel is the human chromosome 15q11, q13, and the bottom panel is the mouse chromosome 7, which is equivalent, or we call synthetic, to the human chromosome 15q11, q13. If you carefully look at it, overall, it's very similar, but you do see some difference there too. So over the time, as Allison alluded to, when I was a student in the Dr. Godash's lab, I created the first mouse model for the Angelman that has been used in many laboratories in the world right now, create a lot of discovery and for the treatment development too. So this is the one I first created. After that, now many additional, so the single knockout are now available, and many people are using, and you just heard that's a rat model too for the UBE3 mutation. We also have two other mouse model, which is we created when I was in Dr. Godash's lab too. For whatever reason, many people did not pay much attention for these two particular lines, which I'm going to highlight here. One is one about 500 kb deletion from the SNRPing gene to UB3A. Exon 2, the coding exon 2. Second line of mice is 1.6 megabase deletion from UB3A to GABA beta 3. So these two mice, you can say, okay, yeah, you're mimicking some Angelman deleted interval, but not truly larger 6 or 5-6 megabase deletion interval. There are a few mouse line, which is recapitulated partially for Angelman imprinting center mutation too. I did not highlight it here because not many people are actually using that one. Which means for people sitting, parents sitting in the audience, that if your kids have an imprinting mutation, that there is a few kind of line possibility that can be used. But if a UPD, I think an imprinting mutation UPD is probably, you would say that's pretty similar molecular mechanism or for that particular type. So the idea is to say, okay, 70% Angelman patient, majority of them, or you are in the audience, are kids that has a 5 to 6 megabase deletion. So it could be type 1, type 2, and type 1 a little bit larger, and type 2 is a little bit small, but that's the major type of Angelman. Now, so far, we don't have a model to recapitulate that particular genotype. So that's the task Allison tried to convince me, say, okay, you have done many other models. Why you take this one on? I say, okay, we actually tried before. When I was in the Dr. Godash's lab, we tried making a little larger deletion, about 2.4 megabase, and using the very old traditional technique from the ES, embryonic stem cell targeting. And we got an ES cell, but we will not be able to get in this particular larger deletion in the germline. And luckily, as many of you know, this new technique, CRISPR-Cas9 technique, is available now. So I told Allison, I said, okay, because this new technique available, I'm waiting to give a try to this challenger test. And we did not really know the time when we do the ES cell why the larger 2 megabase or 2.4 megabase were not able to go germline. We didn't even try to the 6 megabase in the ES cell. It's a part of technical reason because, as you know, human no kind of suggestion that the large deletion is embryonic or the lethal phenotype, as far as we know. But so we would try, but this is a very technical slide. But for many of your parents in the audience, if you're an engineer, you may love this kind of slide. Basically, we are doing a lot of sort of molecular engineering work, and which is highlighted here, which you can see this is the larger chromosomal region where we try to do the engineering to disrupt something. Oh, sorry, I think I make it. I have to go this one. So this first one, it's advanced a little bit fast. So the first thing we do a lot of molecular engineering. Finally, we will say, okay, making the deletion just entirely disrupt the entire 5 to 6 megabase region. The second mice we're trying to do, which let's say, okay, right now you have antisense oligo, you have a lot of other sort of reactivation strategy, you have the UBC3A transgene or protein delivery. So Alice said, okay, let's imagine this way. You correct the UBC3A, but my kids, Quincy, still have 10 other genes, still haploinsufficiency in particular region and other. So what happens if you rescue this one? You still other sort of haploinsufficiency or other deficiencies like a GABA beta 3 or other gene still contribute to her significant problem. So that's a very good question. So that's what kind of motivation is here. Okay, let's make another even more complicated mice. Let's just disrupt this end and also disrupt this end and just leave the Angelman gene entire coding region plus a few hundred kb, which is considered regulatory region, to see what happened. If you see any difference for this particular line compared to the entire deletion line. So that's our motivation. So we've been working really, really hard for here at the Yale Transgenic Core, use the CRISPR technique. So I'm walking through what we have done so far. It's very technical, a lot of work and step by step. So the first step, we're gonna do this, interesting because on my screen, this color all changes. So I'm gonna try to see if I can see this one quick. I thought it's a green, it became white. So we use the technique called harmonious recombination between two special techniques. One is FRT, one is LOXP kind of technique. So we're going to first engineering a side here called the FRT side here. Then we'll plus another one with the LOXP and the FRT. For technical reason, we did it kind of parallel. Then with this assignment, then you see we put a LOXP again first. We second put the FIP here. So kind of sequential step by step, just insert, use CRISPR to here, then we generate the mice, take the cell, embryo, then we make another one, then they combine together. So after so many steps, then because there is a special enzyme with so you can see this is two kind of FIT side here, with a special protein called FLP, which is this mice for. So you can create breeding these two different mice. These mice carry the chromosome 7, carry these two sides. So this mice, it's a protein encoded, carry a protein, special protein called FLP. FLP, when they working together, they were able to acting on these two particular sides. Mediate, we call the recombination happen to this FRT side. Then you'll be by that you'll be able to generate a deletion. You can see this entire interval is deleted, but left only very, very small FRT side. So that's what we do. We did so far. So we were very, very lucky. And the first we got this one, we got a mice, and we would check it. We checked the deletion indeed happened. Technically, somehow, I don't want to go to the subject because many of you are parents. And for the people are scientists here, because as you know, when you're talking to CRISPR, sometimes can target the two different chromosomes. So that can be very, very difficult to handle to making the deletion. So we were very, very lucky. The first targeting we get into this FIT end up on the same chromosome because using the acting flip breeding, we know the deletion indeed happened. So we got a progeny for next generation. As Allison alluded to, we may have the mice with deletion. We do have mice with deletion and this generation, but some of these mice, it's a mosaic, which is a mixture. Some cell is 100% deleted, some cells still not deleted. So we're going to pass this generation to next generation, hope we get a pure germline transmission for the larger deletion. At that time, we will look at the phenotype, hope not for the maternal transmission, we now see the severe lethality. And for the paternal deletion, we may see the lethality. So if that, as we predict, then that will be available for the research community for testing any treatment or understand the pathophysiology. So the second mice, as you alluded to, it's much more difficult to do it, but we are getting there. So as you can see, we have to do this. We have to making the deletion to this end and the deletion to this end. That's what we introduce both FRT side and the LOXP side. LOXP and FRT side is only like about 20, 40 base pair of DNA with very, very special sequence. Then the enzyme or protein can recognize either Cre and GLP protein can recognize this particular site and mediate the recombination. So you can see technically, again, it's engineering mind. If you engineering mind, it's very easy to understand. So you inject, you're targeting here, you're targeting here, you're targeting here. Then we're going to introduce the fourth step. We first cross these mice to the CMV Cre. So we make the deletion on this side first. When we get that mice, you get embryo. So we were going to do the second injection targeting, introduce another FRT side here. Then we allow us to do a second step of breeding using acting FALP for this mice, breeding another mice. Then finally we generate the mice like this, which again, I alluded to the earlier, which you can see the imprinting center for Angelman and across the entire, this is long-coded, antisense transcript or non-coding antisense transcript part of the complex, then come to the UB3 is keep it intact. It's about 700 kb region is intact, but both ends are completely deleted. So that would be very interesting to see if this deletion mice compare the large deletion or compare the single UB3 deletion mice. What's the difference at the phenotypic level? So that's where we are right now. So hope in next year at the time we will have completed the IPSC long project. Then we also have mice to the research community and for you all to investigate whatever interesting you have. So I want to just kind of shout out a few sort of slides for its large teamwork at Yale. First, I thank Allison Brett. As I told you, Allison the one called me, say, hey, take this one on. So I said, okay, let me think about it. And finally, she and I did come to very, very good agreement. So the understanding this is something worth to do it. And definitely as a board of director. And I was shouting out for the Barbara Ballis, and which is she is the chief science officer. I was trying to make a slide and then I couldn't recheck a slide because I have to send out first. And for her kind of continued support for this two very challenging project. Of course, thank you every family, you participant for this project. I know it has been very, very difficult time due to the COVID, but you make amazing efforts to participate in this project since we started and I started contacting you. I will hope and continue to get your support and move this project forward to complete as many as we can. And the Yale Stem Cell Center definitely put a lot of efforts and for this project, and Chai Hong Cho and Jason and two technical persons in particular. And for the Yale Geno Center, transgenic mice and their transgenic core facility working with us very closely for a lot of design, very challenging design. Finally, we will feel so lucky. Every design we made it so far, and so far it's working as we expected. We could be getting to a very, very difficult situation. You have to do it again situation. Finally, it's my own lab. This is a picture took about 2 months ago. We are the first time we can have a lab party outdoor. When shouting out a few people, particularly working the Angelman project, like Cindy Bao and Kun Yung working on the IPSC project, And Taijing is new poster join, and Xiaona is working on the Angelman mouse project. And the Sun-Win actually are listed here. She actually worked on the sister project, the Pedal Volume project. Cassandra is the one and keep all this IPSC consenting in place and contact many of the family for this particular recruitment. So I'm going to stop there. Thank you for your attention. Happy to take any questions.